Hello, welcome to Biology, the scientific study of life. Transcription is the DNA-directed synthesis of RNA. A closer look. Molecular components of transcription. Messenger RNA, the carrier of information from DNA to the cell's protein synthesizing machinery, is transcribed from the, as we see in the figure, template strand of gene. An enzyme called an RNA polymerase pries the two strands of DNA apart and hooks together the RNA nucleotides as they base pair along the DNA template. Like the DNA polymerases that function in DNA replication, RNA polymerase can only assemble a polynucleotide in its 5 to 3. Notice that direction. Unlike DNA polymers, however, RNA polymers are able to start a chain from scratch. They don't need a primer. Specific sequences of nucleotides along the DNA mark where transcription of a gene begins and ends. The DNA sequence where RNA polymers attaches the initiates transcription is known as the promoter. In prokaryotes, the sequence that signals the end of transcription is called terminator. Notice that, terminator. Termination here. Notice that. The termination mechanism is different in eukaryotes. Molecular biologists refer to the direction of transcription as downstream and the other direction as upstream. These terms are also used to describe the positions of nucleotide sequences within the DNA or RNA. Thus, the promoter sequence in DNA is said to be upstream from the terminator, the stretch of DNA that is transcribed into an RNA molecule is called a transcription unit. Bacteria have a single type of RNA polymers that synthesize not only mRNA but also other types of RNA that function in protein synthesis. In contrast, eukaryotes have 3 types of RNA polymers in their nuclei, numbered 1, 2, and 3 or 3. The one used for mRNA synthesis is RNA polymers 2 or the second. The other 2 RNA polymers transcribe RNA molecules that are not translated into protein. Back to transcription of promoter and terminator. Notice also molecular components of transcription. First one here, initiation. This stage, this one is promoter. Initiation after RNA polymers binds to the promoter. The DNA strands unwind and the polymers initiate RNA synthesis at the start point on the template strand. Notice stage 2, elongation. Notice here elongation. This one RNA transcript and this one unwound DNA and this template strand of DNA. Elongation, the polymers moves downstream, unwinding the DNA and elongating the RNA transcripts 5 to 3. Notice that 5 to 3 direction. In the wake of transcription, the DNA strands reform a double helix. Third one, notice that this one represents rewound DNA. Notice here new RNA transcript. This stage termination. Eventually, the RNA transcript is released and the polymers detaches from the DNA. Last one, notice completed RNA transcript. Notice the direction 5 to 3. Notice also the stages of transcription: initiation, elongation, and termination. This general depiction or depictions of transcription applies to both prokaryotes and eukaryotes, but the details of termination vary for prokaryotes and eukaryotes both. As described in the text, also in a prokaryote, the RNA transcript is immediately usable as mRNA, whereas in a eukaryote, it must first undergo processing to become mRNA. RNA polymers binding and initiation of transcription. The parameter of a GN or gene includes within in the transcription start point the nucleotide where RNA synthesis actually begins, and typically extends several dozen nucleotide per upstream from the start point, in addition to serving as a binding site for RNA polymers and determining where transcription starts. The parameter determines which of the two strands of the DNA helix is used as the template. Certain sections of a parameter are especially important for binding RNA polymers in the prokaryotes. The RNA polymers itself specifically recognizes and binds to the parameter in eukaryotes. A collection of proteins called transcription factors mediate the binding of RNA polymers and the initiation of transcription. Only after certain transcription factors are attached to the parameter does RNA polymers second bind to it. The completed assembly of transcription factors and RNA polymers second bound to the parameter is called transcription initiation complex. Shows this figure. Notice that shows the role of transcription factors and a crucial parameter DNA sequence called a data box. Boom. Informing the initiation complex in eukaryotes. The interaction between eukaryotic RNA polymers second and transcription factors is an example of the importance of protein-protein interactions in controlling eukaryotic transcription. Once the polymers is firmly attached to the parameter DNA, the two DNA strands unwind there and the enzyme starts transcribing the template strand. Elongating of the RNA strand. As RNA polymers moves along the DNA, it continues to untwist the double helix, exposing about 10 to 20 DNA bases at time for pairing with RNA nucleotide. Notice that. The enzyme adds nucleotides to the three end of the growing RNA molecules as it continues along the double helix. In the wake of this advancing wave of RNA synthesis, the new RNA molecule peels away from its DNA template and the DNA double helix reforms. Transcription progresses at a rate of about 60 nucleotides per second in eukaryotes. A single gene can be transcribed simultaneously by several molecules of RNA polymers following each other like trucks in a convoy. A growing strand of RNA trails from each polymers with the length of each new strand, reflecting how far along the template the enzyme has traveled from the start point. The congregation of many polymers molecules simultaneously transcribing a single gene increases the amount of mRNA transcribed from it, which helps the cell make this encoded protein in large amounts. Notice back to transcription initiation complex. Notice stage eukaryotic promoters commonly included TATA box, a nucleotide sequence containing TATA, about 25 nucleotides. Nucleotides upstream from the transcription start point by conversion or convention. Nucleotide sequences are given as they occur in the non-template strand. Notice here several transcription factors. One is recognizing the TATA box must bind to the DNA before RNA polymers. Second can do so. Third one, additional transcription factors. Parable, notice the color. Bind the DNA along with RNA polymers, second, forming the transcription initiation complex. The DNA double helix then unwinds and RNA synthesis begins at the start point on the template strand. Termination of transcription. The mechanism of termination differs between prokaryotes and eukaryotes. In prokaryotes, transcription proceeds through a terminator sequence in the DNA. The transcribed terminator and RNA sequence, which means functions as the termination signal, causing the polymers to detach from the DNA and release the transcript, which is available for immediate use as mRNA. In eukaryotes, however, the pre-mRNA is cleaved from the growing RNA chain, while RNA polymers second continuous to transcribe the DNA. Specifically, the polymers transcribes this sequence on a DNA called the polyadenylation. The polyadenylation signal sequence, which calls for a polyadenylation signal AAA in the pre-mRNA, then at a point about 10 to 35 nucleotides downstream from the AAA, a signal proteins associated with the growing RNA transcript cut is free from the polymerase. Releasing the pre-mRNA, the polymers continuous transcribing for hundreds of nucleotides past at the site where the pre-mRNA was released. Transcription is terminated when the polymers eventually falls off the DNA by the mechanism that is not fully understood. Once the pre-mRNA has been made, it is modified during RNA processing. Okay, thanks.